Thank you for having me. So I'm going to talk to you today about my PhD project, which was called Investigating the Influence of Plasmodium Infection on the Human Odour Profile in an Endemic Setting. But the theory that I'm going to talk about is really applicable to all vector-borne disease. I started with an introduction on olfaction, but Gabriella Gibson gave such a lovely one earlier, it seems slightly redundant. I'll go through it quickly. Olfaction is extremely important to insects. They use it to go through a number of their life cycle stages. It governs some really important life cycle stages, including finding nectar or finding a host. And the more we learn about olfaction and the processes that underlie it, and particularly the volatile compounds or the chemicals which are involved, the smells which insects are seeking, then the more we can exploit these in traps for monitoring, but also for control. We have traps which use the nectar-seeking behaviour of insects, called toxic sugar baits. Again, Paul already mentioned this. And these are useful against mosquitoes and sandflies. These are for control. We have traps which exploit the sex pheromone or arrestment pheromones in ticks or aggregation pheromones in bedbugs. We have traps which exploit oviposition behaviour, particularly in mosquitoes. And these use the oviposition cues, including synthetic oviposition pheromones, for surveillance and for control. And then perhaps most of the literature is about host seeking. So again, the tsetse fly, a really classic example, but we're exploiting the cues that these insects and arthropods use to find their hosts. And that's kind of where my work is focused. So I really want to emphasize this, it's not just for surveillance, but for control, and you can have a measurable impact on the transmission of disease. In 2016, the results of the solar mal study were published. And in this study in Western Kenya, the investigators installed what they called odour-baited trapping stations outside houses. And these were powered by solar panels, which also give people electricity inside their houses. It was a cluster randomized controlled trial, and they rolled out the intervention across a whole island. And throughout this intervention, once they started measuring malaria prevalence and also mosquito numbers, they crashed the population of mosquitoes such that prevalence in the intervention cluster was significantly reduced relative. Where are we going? Yes, that's the intervention, and that's the control cluster there, clusters there. And it was malaria prevalence was significantly reduced. So this is a fantastic finding. To train. So I was going to have something else there. So blood-seeking insects take a meal to get enough protein in order to lay a batch of eggs. In the case of mosquitoes and malaria system, during this process, they might take up the gametocytes. These are the transmissible stages of malaria, and then they'll become infected, and then they can transmit the infection onwards. Again, they use a variety of cues to approach their hosts, and importantly, odour is used at all distances until the close range where heat, humidity and vision is used as well. So the body odour, which I'm talking about, are chemicals which emanate from your skin. So that's the chemicals that you produce yourself, so sweat, but also it's the metabolic products of the microflora which live on your skin. To change track slightly, parasites are known to manipulate their hosts in order to maximise transmission. In this picture, an ant is infected with a nematode parasite, which is inside its abdomen. That ant's abdomen is normally black. And it's made the ant climb up a tree, stick its bottom in the air, and it's gone bright red, and it's easily detachable. Why is it doing this? The ant is inside a cluster of red berries, so it looks just like a berry, and the bird comes along and it eats the berries and it accidentally eats the ant, and that parasite is then passed on to the next host. So this is the first documentation of parasites causing things to pretend to be fruit, which I think is quite cool. Why am I talking about ants that pretend to be fruit? We wondered if this is happening in the malaria system or in Plasmodium. So as I said, the host cues are really important to the mosquito coming to bite the person. And there is some evidence that suggests that people who are infected with malaria attract more mosquitoes. They're more attractive. Why are we interested in this, or what's the outcome of this? Well, as I mentioned in this study, the lure that was used here replicated that of a healthy host. It basically is like a people smell, a synthetic person smell. If people who are infected are more attractive, then we could improve that lure and we can make an infected person smell. So this might become a more efficient tool. So the aim of my study was to determine whether infection of the human by Plasmodium species alters the odour profile of humans in a way that influences attractiveness to the mosquito vector. And I did this through two objectives. First, I did a chemical screen of the odours, which are produced by individuals who were infected and uninfected, and then I did an entomological analysis, so I looked at the mosquito response to those compounds, to the same odour samples. The study was based in Western Kenya, here on the edge of Lake Victoria. We went into schools and we recruited 5 to 12-year-olds. We tested them using point-of-care measures, so film and RDT. We took a number of other measurements, and then we went away, we read the films, and then the next day we came back and we took odour samples we tried to get a negative individual, someone with asexual stages and someone with gametocytes at each sampling effort. We then treated them with antimalarials, and we came back 1 week and 3 weeks later and repeated the whole process. In total, we sampled 56 individuals, and with repeated measures, we had 117 odour samples. So as I said, the odour profile was sampled from the foot using a technique called air entrainment. So that just captures the chemicals that are coming off the foot. And then I analysed them using gas chromatography. It's a way of separating out the constituent components of a complex mix, basically. So you inject your sample here, it passes through a really long column, and the components separate according to their characteristics. And then it's detected, and the output is a trace. And a peak here represents a chemical, basically. At the same time, we did the parasitological diagnosis. Like I said, we used point of care in the field, so that's the films and the RDT, and then we did more molecular testing retrospectively. In the end, I ended up with 101 positive samples and 16 truly negative samples, negative by molecular measure. Of those positives, I had 20 who had gametocytes by microscopy. I had 32, which had what I call low-density infections, so that's less than 50 parasites per microliter, and that's approximately what a microscopist will be able to detect. And then I had a bunch which were over 50, so I'm calling them my higher density infections. And the figure over here just shows parasite density in these 3 positive groups. So the middle group is the high density infections, and you can see everyone's above 50. This is what the line is. The lower density, everyone's below, and those with microscopic gametocytes tended to have a high density infection, as you would expect. So I analysed this data using multivariate statistics and also using an R package called Multiquant. As I mentioned before, the output of a GC is a trace, and a peak means a compound. So here, I've overlaid samples from lots of different people, all showing one compound. Each one is a different compound. And I've coloured the trace according to the parasitological status of the individual. So those with high density infections are purple, pink is lower density, and then the green is negative. And you can see for these 3 compounds, those with the high density infections really are producing more of those compounds, and it seems to be density dependent, so the lower density individuals are still producing more, but a little bit less. I then took the actual amount of compound, I normalized it to the total amount of compounds that were collected from that individual, and I did multivariate, no I didn't, I did mixed models to work out whether there's a significant difference between the individuals in the different groups, and I found for these 3 compounds, significantly more was produced by those with higher density infections and also often with gametocytes. Because of this seemingly density-dependent effect, I took my individuals and I divided them up further, just so instead of just lower and higher, I did quartiles of low, medium, high, and high. And again, that's the parasite densities in these groups. And then I looked at the compound production, and you can see for each of these 3 compounds, you've got this really nice increase in compound production according to the parasite density that the individual had. You lose the significance, but that's largely because the difference between the groups is a lot smaller. I should mention those weren't the only 3 compounds that I found. I found a number of different ones, and here in the middle, again, it's a GC output. It's just 2 example traces, so 2 individuals, and the one that's coloured red, somebody who had high density parasites, and the green is a low density parasite person. And the arrows are my compounds of interest. So these are, well, infection associated compounds, ones I found to be produced in higher amounts by individuals who had malaria or high density parasites. And I think this trace nicely shows that the compounds which are being produced in greater amount are actually the ones which were already highly produced in the odour profile. So this is quite a distinct signal. The figure at the end just shows the same thing. So again, I've divided my individuals into those categories, but And each bar represents 100% of all the chemicals in the odour profile, but I've coloured my infection-associated compounds at the bottom there. And the grey is about 170 chemicals. I just didn't bother putting in lots of lines. So you can really see that the infection-associated compounds, the compounds that are changed on infection, make up a large proportion of the odour profile. So if you look at the total emissions for individuals, so we thought, well, maybe just everybody produces more of everything when they're infected, but actually, in terms of total amount of compound produced, there was no significant difference between the groups. Possibly a slight increase in the higher density group, but you might expect that if one or two compounds are produced more highly. So the next thing I did was an entomological analysis. So I took the same odour samples and I did EAG, GC-EAG. So if you're not familiar with it, it's the same GC work here, and you get your trace, which is the top line here. And then the sample's actually split, so half goes to the detector as per usual, and the other half gets blown over a dissected mosquito's head. And we monitor the electrical impulse that's generated by antennal nerves firing. So when antennae detect something, the nerve cells fire, generates a tiny electrical impulse because of the depolarization of the nerve cells, and then you amplify it. And what you get is this bottom line, and where there's a depolarization, you get a dip. So that's a response, that's a response, and that's a response. And you can take your odour sample, and then you do this, and you can work out which compounds are interesting to mosquitoes. So I blended my odour samples. I used slightly different groups because we really wanted to kind of push this idea that maybe it was the transmissible stages, it was the gametocytes that were causing changes. So I pooled odour samples from individuals with gametocytes. Subgam is a low density of gametocytes, just with asexual stages, and then individuals who are negative. And I find that actually mosquitoes responded always to the same compounds in those groups. There wasn't any qualitative difference between them. So they approximately responded to the same compounds in each treatment group. However, amongst the compounds that they responded to were these three compounds that I showed you earlier. So, taken together, we know that several compounds are found to be produced in significantly greater amounts by individuals who have malaria relative to those with lower density infections or with no parasites, without an overall increase in volatile emissions. The production of some of them appeared to be tightly correlated with parasite density, and several of these were found to be EAG active, so they induced this antennal response. And that doesn't necessarily tell us what the behavioral, well, it doesn't tell us what the behavioral response is, so it could be an attraction or it could be repellency. Although we have some preliminary data from our collaborators that these are attractive compounds. And also, are these compounds directly produced by the parasite, or could it be some sort of inflammatory process that underlies this? And does it mean, like, is there a repercussion in the field? Does this actually change the transmission of malaria? So thanks to everybody who helped on this project, and thank you for listening.